July of 2020, an alleged sexual assault was reported to the UTS Forensics Laboratory, as forensic biologists were tasked by Constable Jane Devey and James Brown of the Sydney Police Force to search for evidence of human biological material on each of the exhibits submitted, in addition to determining whether DNA from either the victim and the suspect could be recovered from any of the items. A case file was maintained throughout the course of the examination and DNA profiling process, and from this we have produced our expert opinion court report. Constable Jane Devey attended the scene where Mary Wang alleged that John Smith sexually assaulted her. She claimed that he used her mobile phone before the alleged assault and that she was injured during it, causing her to bleed. A drink bottle and cigarette butt possibly belonging to or used by the suspect were also recovered from the scene. Miss Wang consented to a vaginal swab and a reference buccal swab being taken. The victim's pants appeared to have a possible semen stain and pubic hairs present. Constable James Brown arrested John Smith at his place of residence, also recovering a white shirt with apparent blood stains. These were the main subsource propositions we considered during the analysis. The exhibits were examined and traces were collected and swabs were taken. All traces underwent DNA extraction, quantification, and amplification. The victim's vaginal swab was divided into two components. Following this, the samples underwent capillary electrophoresis, a process to separate and detect target DNA sequences required for the creation of a DNA profile. With this complete, analysis took place using Osiris and RStudio software in order to produce likelihood ratio.
The sequences of DNA used to create a DNA profile are known as short tandem repeats. The Royal Society best explains that mutations that affect the number of repeats are relatively common. So within a population, there are usually several different versions of the DNA. How frequently a specific allele is present at a certain locus can vary between ethnic groups. We use this information to determine the likelihood ratio of two unrelated people in a given population having the same or similar DNA profile, as outlined in Collins and Morton's 1990. It was crucial in our case that we determine whether the suspect's DNA appeared on the victim by chance or random match, and vice versa. Likelihood ratios were calculated for each exhibit for 3 subpopulations. The likelihood ratios of both reference buccal swabs and the suspected pubic hair are shown on the slides now. The remaining exhibits will be addressed later on.